this could be good. There we go. All right. Yeah, no, it's a good one because essentially what they show is if you concatenate runs together, this started with resting state, the functional connectivity goes up over time.
Right.
I thought that's, they thought when they first saw this, they thought, oh, that's interesting. It's the effect of the drug they were giving or whatever it was. And then they realized, wait, when we look at ABCD data, HCP data, UK Biobank data, everybody does the same thing. This is not everybody, but 80% of the people they looked at. So it seems to be arousal based. So basically, the longer you're in the scanner, the more you drift towards some new plateau. And it seems like the way that they pull out the physiologic noise is using Blaze's rapid tide. So this is nice because it's a model, right, that we understand. So although it doesn't work mathematically this way, you can conceptualize this as being like seeding in a carotid and mapping anything that goes up and around the circulatory system and comes back to the veins, to the jugulars. By definition, then systemic, right? So it hasn't got a local control. So body wide. And we know there are at least three good candidates. So it's probably all three. Non-stationary oxygen saturation on the arterial side, which Toshihiko Aso has shown. That'll give you a bold effect. Non-stationary CO2 because of the vasodilation. That's a big one, which Power and others have looked at, and is probably the biggest component in the global mean signal. And then anything that's blood pressure related, Meyer waves, anything autoregulatory, right, which would also be very, very slow. And all of those have some coupling with arousal. So they just pull this out and they show that the gradient in the run-to-run functional connectivity goes down to almost a perfectly flat line. There's a very shallow gradient left, which might well be neural, right? That's super interesting. But you have a principled way to pull stuff out, which I think is a super, super cool thing to do. I'm hoping everybody does this soon.
When you say seeding in the carotid, do you mean actually?
Conceptually. So the way that they do this is like a flowing regressor. So imagine if you take at time one, you drop the seed in a signal in the carotid, and then you ask the question, +1 TR from now, where does that waveform end up? And it's moved up and it's now here, it's circle of Willis. TR +1, now it's diverging in the brain. And you can literally track anything that flows. It's as if you're riding along with the blood and it's going right through the system. The way they did it at first, they seeded in the superior sagittal sinus and went backwards in time as well as forwards in time. And then with Russ Poldrak's myconectome data, they showed that him on and off coffee, which was a nice control, 100 odd data sets, that they could get the same results seeding in the carotids because they had enough coverage to get down to the carotids. So they could go only forwards in time. And then for mathematical convenience, the way it works now is if in rapid tide is that they just take the global mean signal, that gives you a starting regressor, and then you allow it to just go from the global mean towards each voxel, and it just goes forwards and backwards in time, maybe 5 to 10 TRs, and it converges on the voxel-wise local appropriate lag. So it's super, super fast to converge. So, it's a nice principled method.
Like a kind of phase matching thing. Yeah. Okay.
Yeah. Cool. Yeah. So there's that's a mathematical way of doing it, but just because it converges more quickly. But you could do it with superior sagittal sinus seed, it will give you the same result. Very slight differences. And some of the gotchas that if you want to read more about this, is people who breathe very regularly, like me, are bad candidates for this because you can get a lot of autocorrelation in the time series. All you need to do to break that is one sigh or one brief breath hold, and that breaks up that periodicity. Most people do that by definition. But Bayes has some nice principled ways of what he calls despeckling, where you can, if there's some ambiguity, you can use your knowledge of flow, that the whole blood flow can't be more than normally 10 plus seconds, 10 or 12 seconds in a normal brain. Then you can decide which of the waveforms is more likely to be the right one. Or you can just look at next nearest neighbors and ask the same question. Well, what did they do? It's unlikely it's one voxel, it's going to be an outlier, right? Because they're all being influenced by the same through space effects. So that's really nice. And I think that, given that global signal regression has so many statistical issues on its own, let alone everything else, that just doing a regressor, a lag appropriate regressor, I think is a very easy next thing for people to do.
So these traveling waves you get throughout the brain, they're happening with every cardiac cycle, or are we talking about?
No, typically much slower. So the low frequency oscillations are typically below about 0.1 hertz. So it's at the speed of things that are flowing with the blood. So think mean arterial blood pressure.
Okay.
Right. Things that are sort of slowing seconds to tens of seconds.
Okay.
Like arousal, which would change the blood pressure. Changes in PaCO2. I had to hold my breath inadvertently for 10 seconds while I'm thinking about something, and there's an imprint of CO2 that's going to now have to course right through the vasculature. And an oxygen saturation. So blood gases are the big ones, I think.
Okay.
Biggest. I suspect autoregulatory phenomena are second biggest, and maybe there are some other as yet unknown phenomena.
Okay.
But it's things that are you can think of it as a modulator of bold. So anything that's modulating the global bold signal through the vasculature.
Right.
Yeah. Yeah.
And it's low frequency stuff. So that's, I mean, there's stuff we can actually address and remove. Oh.
Yeah. And we are doing that. I mean, that's what they've done a pretty good job in that paper. It's pretty thorough. They've compared against global signal regression and fix and other things, and they just show that none of these, the global signal regression throws things out overly aggressively in one respect, but under aggressively on a local basis.
Yeah.
So that's a bad thing to do, right? We knew that, but there is an alternative now. You just do rapid tide instead, and it's all in the Erdogan 2016 paper. People just got to start using the tools. I suspect a lot of this is just workflow. If rapid tide was bolted onto fix or something from the grid, then people would just use it. So maybe that will happen.
And is it plug and play? Is it just
as far as I know, I mean, it's all on his GitHub and everything, and he's available to help people. There's a few people doing it. I know Ahmed Khalil, a few other people have picked it up and have run with it pretty well.
That's great. I've spent quite a lot of time trying out different methods of noise reduction because I'm always trying to pull crappy small effects out of crappy small data sets. Because the kind of the stuff that we do, with the psychedelics and that kind of thing, these studies are so difficult and so expensive to do. Just in terms of, it takes 2 or 3 years to set up one of these clinical trials and get the drug and get all the right permissions and get the license from the Home Office and all that shit. And then, it costs a lot of money, a lot of staffing time, doing the therapy and so on. So, we always end up with 20, 25 subjects if we're lucky, So I'm always keen to try and get the most out of these data sets that we have. And, I tried things like fix and aroma and, manually denoising stuff using ICA analysis and things like that over the years. And I've never found there to be a magic bullet that works for everything. Some things seem to work okay on some sets of data and other things work okay on other sets of data, which is really frustrating. But maybe the rapid tide could be the thing. I mean, I like the sound of it because it's very principled.
Right, exactly.
It's theoretically grounded and sounds plausible.
I think that for me, that's the most important point. Independent measures are nice. We had chest motion, you could do RVT, etc.
Yeah.
There's a model, right? It's a sort of a forward model. But this has been validated now. Yunji, Tong, and Blaze have been working on this since 2010, right? They've been around the block with this stuff. They have pulse leads with near infrared, etc. So they really understand this stuff, and it's robust. And I think now there's enough prior literature and prior experiments that I would trust a data-driven method already here, whereas I might not for something like arousal. You know, if you're going to tell me I've got a model to pull out arousal from fMRI, well, okie dokie, maybe, you know, I want to see the validation there, right? But I think there is enough here, and I think we have enough of an understanding with CO2, especially, that we know what it's doing, and we can easily break it too. You can test this. The nice thing about this too is if you don't believe it, all you've got to do is take your same subject and a small subset of them, have them do a breath hold task.
Right.
Even just a simple one, just have them sigh on a cue when you tell them to. That will be enough to imprint something in your data set. And now you can ask a rigorous question of your data. So yeah, I think that's good. Yeah, the heart rate stuff is the other one I've been pondering a lot. And I think I would guess, my guess right now is that fix or anything that's doing like AComp core is probably doing a reasonably good job of pulling out heart rate pulsatility effects, because most people are using the CSF, the ventricles, and that's sort of almost an amplifier of global pulsations in the brain. So I would think that that's doing okay for heart rate and heart rate variability globally, but the problem is it's a nonlinear effect because it's going right. So would we end up with potentially spurious small local effects that we could misread? Yeah, I suppose that's true. But it would be highly unlikely to be reproducible across individuals.
Yeah.
So maybe that's the trick.
Yeah. I mean, I got to the point a few years ago with this stuff where I almost gave up, because I was trying to find something that would work reliably on lots, you know, on any arbitrary set of data, you know, and I couldn't really find anything. Yeah, we tried. I've got a bunch of data that I haven't written up from years ago, which is really stupid, but I'm going to get around to it. But we did four different tasks in the scanner plus resting state, and then we analyzed it in 4 or 5 different ways, 4 or 5 different motion correction, no, not motion correction, physiological noise correction things. And we had subjects doing 2 scans a week apart as well, and we're looking at test reliability. And my idea was, okay, if we find the best correction method, physiological noise correction method, that gives us the best boost in reliability across these 4 tasks, that will be the one that we'll use on all our pharmacological studies, because you're always worried about test reliability when you're doing, you know, a placebo scan and then a week later doing an active drug scan, right? And it turned out, well, you know, one of the methods worked best for one task and another one worked best for another task. And I was like, Christ, so what do I do now? So eventually what I've basically settled on for the last few years is a kind of comp core power type method.
Which I think everybody's settled on now, and everyone's still arguing whether global signal regression or not, but that's it. So everyone's doing fix or AComp core, minimally processed stuff.
Which again, I kind of like, because it's principled. You know, you're using signals from the white matter and the CSF, where you know there's no, well, particularly in the CSF, you know there's no bold activity. Anything there, you know, must be noise, and it doesn't really matter what the source of the noise is. You know, whether it's physiological or thermal noise from the scanner or head movement or whatever. It's like, this is noise, we just want to get rid of it.
Yeah. So I think, the CSF, I think, will probably do a pretty good job of heart rate effects.
Okay. Oh, that's good
the white matter, I don't really know. I mean, probably, you know, it'll be inappropriate though, because the white matter vasculature is so much slower than the grey matter. So I don't know what you get really out of the white matter. There'd be a question that people would do. Just turn the white matter correction off. Does it make a big difference? Especially if you're going to then do something like global signal, mean signal regression anyway, it probably doesn't get you very much. But we probably don't need to worry about that anyway, because if you just use rapid tide, whether the white matter correction is doing anything or not, as long as it's not adding any color to the data, who cares? If it's not doing much, all right, leave it in, just in case. Yeah. So yeah, I think there's and that's the thing, you know, I'm trying to be optimistic here. You know, I think we just need a few more people to do a few more principle, validation studies to sort of move the needle a little bit on, the blood gases. Now I think Blaise has done a good job here. I think now the next thing on my radar is arousal. That's a big deal for drug studies. So I think that's sort of where I'm coming at this from, because we've got our psychedelics folks here too. It's, it's the first time I've really had to look at effective drugs at all, short term or long term, and there's a lot of
nightmare.
Nothing stationary, nothing.
Yeah.
But then that's sort of why I'm an experimental scientist. I look at this and say, right, knowing what I know today and trying to anticipate where this field might go in the next 2 or 3 years, which would be the sort of time frame for when people will be writing those data up.
Yeah.
What do I think is essential measurements, independent measurements, or ways to acquire the data, because that could be equally important. What's must have, what's nice to have, and what who cares, right? And I think right now, chest motion, nice to have, but probably not critical for me, because I think I've got principled ways to get at the data. Where it could be useful though would be if you have a question, if you just collect those, why not collect it if it's easy, right? And then if there is a question about, well, what was the person doing? What were they holding their breath for 30 seconds or inadvertently? We go back and look at the gold standard data. So why not collect that? Pulse ox the same, right? And I think for things like drug studies, I would want to ask the independent question. The first question I would have is the heart rate and the heart rate variability the same on and off the drug? Is there a systemic difference between these two conditions 1 week apart, or this bunch of people and the controls, right? If they're on placebo and their heart rate variability is 20% lower, okay, flag. It doesn't necessarily affect the data, but it means that I go into the analysis with a little bit more circumspection, right? And I think that's all we can do.
Yeah.
So what about arousal? What are you guys interested in trying to measure arousal? Is this something that you're going to try and get a handle on, eye tracking, say?
Possibly. What, doing like pupillometry?
Yeah. Or insert your favorite arousal measure here, GSI.
Yeah, sure.
Fidgeting index.
Well, I mean, for some drugs certainly, but I mean, the thing that I've been doing quite a lot of recently is actually sex research as well. So I don't know if you've seen those papers.
I have, yeah, God, it makes studying drugs look easy.
I mean, the sex studies are hilarious, obviously, right?
Yeah, what doesn't change?
Well, yeah, exactly. So we're showing people an 8 or 10 minute pornographic film in the scanner, right?
Yeah.
And we're recording heart rate and so on. And for the guys, we've done it in women and men. For the guys, we actually had a little device that we attached to them directly to measure their
a part of their anatomy.
Yeah. And guess who had to try that out when we first got it, of course.
And then that didn't work, so then you had to get someone else.
Yeah, I've been married for years. It's basically just for pissing now, frankly. You're still recording this, aren't you?
Shit. Yeah, sure. English blokes, so everyone's gonna be like, yeah, Londoners. Oh, well, one of them's Australian. No.
Yeah. Oh, where were we? So yeah, I mean, obviously, arousal of various kinds is a big deal in those studies. Yeah, for sure.
Do you worry about hematocrit in those situations? Just asking. Does hematocrit change if you have a, you know what?
I honestly have no idea.
Might be important to find out.
Yeah.
And then blood pressure will be a good one. So I'm hoping to get some information from Blaise soon. I just tried the stupid experiment because, blood pressure non-invasively, very, very difficult to do. But, and you can do pulse wave velocity. So we happen to have a couple of old pulse oximeters. So I just tried it on one finger and this left finger and left toe. And there's a good 100 to 150 millisecond difference in the arrival time of the pulse.
Right.
So the question now is,
That's
does
cool.
does the arrival time change with blood pressure? And I can measure that outside the scanner. I don't need to do this inside the scanner. So I'll just use a cuff as a control. So if that starts to look like it's working, then we have a relatively easy way of getting relative blood pressure, which is all you and I care about. You just want to know
Sure.
what time course.
Yeah. Oh, that's very neat.
Simple, because that's
Very
how
good.
the brain works.
Yeah, no, sure.
I'll keep you posted on that.
Simple
But yeah,
measures
I
of
think blood pressure, you'd want to know what blood pressure was doing somehow, right? And
yeah,
I would think about hematocrit too.
yeah. Yeah, I mean, we got um cardiac and respiratory data when we were doing these subjects. But we haven't really done much with it, to be honest. So we've done basic analysis of all these data. But we did our standard comp core like approach, and didn't really do much with haven't done much with the cardiac and respiratory data. And I've been thinking for a while, it'd be really good to look at heart rate variability or whatever other sensible measures we can get, and see how that affects what's going on in the brain. But
Interesting.
might have to
Yeah.
wait for a master's student, the right master student to come along, or something like that to do that.
Maybe start them on something easy like psychedelics. Don't put them on the
Well,
same straight away. boy, that's
yeah.
difficult. I wondered too, something you could possibly look at, an easy low-hanging fruit. I wonder if you would see a difference in if you have some sort of IR measure that looks at total hemoglobin. I mean, well, I guess pulse oximetry is looking at total hemoglobin, isn't it? But it's not giving you a ratio of oxy to deoxyhemoglobin in your standard pulse ox, right?
No.
It's just doing whatever the wavelength is. I do wonder if you would see a change in the total absorbance in a finger when your man there has his erection, just because of potentially a change in blood volume. Blood pressure might compensate. I don't know what yeah, because you can imagine the blood pressure might elevate. It might compensate somehow. But
Yeah.
might be
Don't know.
interesting just to look at the peripheral physiology. Don't even think about the brain for a sec and just try and understand that.
Yeah.
That's how Blaze got into all of this stuff was functional near infrared. They were recording FTIR on the brain, and then they had a finger, and they're like, "Wait, I see the same thing in the finger that I see in the brain. How can that be?" Right?
That's cool.
Yeah. So maybe just starting with the physiology first wouldn't be a terrible idea. Well, interesting. So how do you approach the issue of psychedelics from, like, when you're doing acute versus chronic?
Yeah.
What do you think about differently when you're trying to do these? And I'm thinking acute, obviously a dose 1 week apart versus microdose, low dosing over the course of a year, maybe with some other therapy, and then looking at brain differences.
So I mean, we've done kind of two kinds of studies. So we haven't done an acute study for a very long time.
Okay.
Well, I mean, the Imperial guys that I've worked with a bit, they've done some. I haven't really been very involved with it. But we did do, I mean, about 10 years ago, we did do an acute MDMA study, and MDMA obviously has big effects on all kinds of physiological, measures. What we've tended to do recently is more clinically related kind of therapy trials, where we scan them at baseline, and they then go and have their psychedelic therapy, and then we scan them a few days after, or maybe a week
It's
after.
mostly like depression cases, PTSD. Yeah, depression.
We've depression was done a few years ago. Most of that's been published now. We've just finished a study in anorexia patients, actually.
Oh, wow.
Yeah, psychedelic therapy for anorexia. The Imperial guys are doing chronic pain patients as well at the moment.
All right.
And
So for any of those, what do you do for behavior, sort of outside the scanner behavioral changes? What kind of standardized tests are you looking for? Like anorexia, if they're going to gain weight, that's going to change things. if they're depressed and they start getting more exercise, that's going to change things, right? So
Yeah.
what kind of external non-MRI measures are you recording and why? What kind of studies? What kind of
Well,
questionnaires?
I mean, these guys are really intensively intensively studied.
Gotcha.
So it depends a bit on the disorder. Um, but I mean, let's talk about the depression studies, I guess. So loads of different clinical and kind of emotional related questionnaires. They started using EEG measures in the recent studies as well, actually,
Good.
which is interesting. So they're able to get EEG during the actual dosing sessions as well.
Oh, wow. Nice.
A little bit, yeah.
So you get some acute, potentially acute effects there. That's nice.
Yeah,
All right.
exactly. And they've developed this well, people in New Zealand actually, and they've come over and taught us how to do it. They've developed this method for getting it's a bit, I don't know, for me it's always seemed a little bit indirect and a little bit of a stretch, but they do this visual task, and the changes they see on this visual task are supposed to relate to long-term potentiation of the neurons, and so they're using it as a marker of neuroplasticity because psychedelics are supposed to increase kind of flexibility and open this kind of window of neuroplasticity.
Does the
So
performance enhance? So not so much, you could measure the activity during that, right? But does their performance get better too?
yeah,
Yeah. Okay. Which is undeniable, right?
yeah, yeah.
That's bulletproof.
Um, but the point of this task is that it's supposed to be an index of how plastic. So it's a kind of like a habituation type visual. It's quite simple stimuli. It's like
Yeah.
visual gratings.
Yeah. So
But
showing your perception or your awareness or whatever.
yeah,
And
yeah.
then this is doing an fFMRI as well. So you have
No, this
no.
is this
Okay.
is this is separate.
Okay.
No.
Okay. Just
Yeah,
purely behavioral. Okay.
yeah, yeah,
Good.
yeah.
Well, that's nice because that's a validation that potentially could be migrated into the scanner once you get a good handle on any
Yeah,
other confounds in the scanner. That could
yeah,
be nice.
yeah. I mean, I'm really keen to try and derive, neuroplasticity type measures from fMRI data as well. And I mean, I know there's been a lot of stuff proposed on how to do that. I think it's all a little bit indirect and not terribly convincing, to be honest. But
This is you talk about fMRI studies or just behavioral studies more generally.
well, I mean, no, I mean, there's lots of behavioral and EEG markers of neuroplasticity, if you like, or
Uh-huh.
proposed markers of neuroplasticity. There are some MRI measures that have been proposed as well. I mean, you can do diffusion work and look at
Long term.
FA and microstructural changes and things like that.
But that's
I'd
months to
like
years.
Well, yeah, you
Okay.
say that. No,
Okay.
you say that. No, I mean, I think
Okay.
we've got some data coming out soon showing
Okay.
changes in diffusion after 4 weeks, after
Okay.
one psilocybin trip.
Wow. Oh, okay. Wow.
Yeah.
Are
Um,
you gonna I'm gonna ask you a reviewer like question. Does it stay? Do you just try and see if it washes away after time? You try and consolidate it?
we don't know, although there's a preprint that was out a while ago, not from us, from Nico Dozenbach's lab,
Okay.
that?
Yeah.
Yeah.
Mhm.
Uh, where they did their kind of very dense sampling thing, where they had quite a small group of subjects. I think it was 7 or 8, but they scanned them 17 or 18 times each,
Mhm.
and they did acute psilocybin, and then a day later, 2 days later, a week later, month later, so on and so on. So they were looking at this effect you get with psychedelics, which is general disruption of resting state networks and kind of randomization of the patterns. And they found persistent effects, I think, over about 4 to 6 weeks. But then by 6 months, it had kind of gone back to gone back to basic.
Washed this out. Okay.
Yeah,
And that was with fMRI, not FA,
yeah,
not fusion.
yeah.
So yeah,
Definitely.
okay. Well, that's super interesting. And I suppose the next thing is mechanism, right? What is causing the FA change? Is it really, fiber density, or is there some cellular, some cellular change, or even a CBV change?
Yeah, I don't know. Good
Cool.
questions.
Yeah,
No
yeah.
idea.
Good.
Yeah.
Well, first thing is have something that's reliable to measure, right? And then
Mm,
ask mechanistic questions. That's super interesting.
mm,
What
mm.
other neuroplasticity measures then do you think people could be looking at? You've got your visual sort of spatial awareness.
Yeah.
What else do you think might have legs?
Well,
And that
I
that
mean,
that's the spatial one that you're talking about. There it has the FA changes, right? This,
yes.
yeah. Okay. Cool.
Yeah.
It's nice and simple.
Um, well, I mean, this is a bit out of both of our wheelhouses, but we are doing a bit of work at the moment where we're using PET. Um, so there's a PET ligand called UCBJ, which is which binds to the synaptic vesicle 2A protein, which is a protein which is expressed in almost all synapses.
Oh, nice. Okay.
So it's a marker of synaptic density, basically.
Any type excitatory inhibitor.
Any type.
Right. Okay.
Yeah.
Sweet.
Yeah.
Okay. Because it's
So we've done a little bit of work. It hasn't worked out fantastically. So I know there's only a few labs in the world that are using this tracer in PET studies. So there's people at Yale. And I know they've tried this as well, and it hasn't worked out for them. They haven't seen any changes after a psychedelic experience.
okay.
I think they used ketamine as well,
Okay.
which is, people argue about whether ketamine is really a psychedelic or not,
Right. Okay.
because it's got a very different pharmacology to psilocybin and LSD and things.
Mhm.
We've tried it as well in London, also with ketamine, and didn't see very much either.
Right.
Um, the guys in Copenhagen, who have done most of the kind of psychedelics PET stuff that's happened so far, I think they're also trying it at the moment. We're trying it again with DMT, which is a proper psychedelic, if you like, a proper 5-HT active psychedelic. We're actually doing it on a combined PET MR scanner and getting
Nice.
fMRI data at the same time, which is cool.
Yeah.
Yeah.
Wow.
So that's exciting. We're only part way through that at the moment. But probably, I mean, problem with PET studies is they're just so expensive.
Sure.
Um,
But
uh,
somebody's proven that this will work. If you get a change in synaptic density, you will see a
so
take in
it
this.
works. It works in animals.
Yeah.
So
Okay.
it works preclinically in rats and monkeys and stuff. People have showed that you get changes in synaptic density after
Oh,
psychedelics
okay.
with this tracer, but it's never really been shown in humans.
Okay.
Uh,
Well,
so yeah, we think it should work.
are there any other ways you could change synaptic density without using drugs? Without using these kinds of drugs? I'm just trying to think if all you're trying to do is, so first of all, prove that you can do it in humans. Might be the easiest
Well,
way.
so I mean, there are things you can do, yeah. So you can do kind of adaptation type studies. So you could look at the motor cortex at baseline, and then you could do things like, people have done this a little bit. You could kind of bind somebody's fingers up for a couple of weeks, and
Oh,
then look at how that changes the finger representation in the
oh,
in the opposite
okay,
side
okay.
motor cortex, and you
Oh,
you probably do see changes when you do things like that, or
okay.
put a patch over one eye for a week
Yeah.
and
So
and
visually.
that kind of thing.
Oh,
Yeah,
okay. Right. This is not a leap of faith. I mean, you're confident that at least beyond some level of synaptic density change, you will see something. So it's maybe a
yeah,
sensitivity argument. Brilliant.
yeah,
Fantastic. I
yeah.
look forward to seeing that. That's really
Mm.
interesting. Super cool.
Then we have the issue of how to try and marry the PET and the the MRI data together, of course.
Well, yeah, one step at a time. But I think if you, yeah, but if you just get the PET result, that would be huge,
Yeah.
right? And not everything has to be MRI. You know, the MRI isn't the answer to every
Sure,
question.
sure. Yeah,
But
I,
that
I
just means
know, but
I,
and I know it's partly because it's the environment where I work. So the center where I work, we have PET and we have MRI, right?
yeah,
So obviously, at least to me, it's always nice to try and build bridges between those two things,
absolutely, absolutely.
And but I think it's cool. You have the molecular level data with the PETs, particularly in drug studies. You have the molecular level data from the PET side, and then you have a kind of functional physiological level. And I think it's cool to try and build that bridge between them, because
Yep,
the molecular level is where the drug is
yep.
having the effect clearly, and it's then kind of feeding forward into
No, I think you make
some
this
kind of functional effect.
takeaway for viewers. This is to me, this is like this is exactly the whole the the mens rea for these kinds of studies, right? Is that you can't answer everything with MRI alone. It's not just observational or MR astronomy, as Blaise likes to call it, right? Sometimes you need to poke the universe to find out what it's doing, right? You can't just observe. So, whether it's PET or some other intervention, then I think this is either another way to see the same thing that doesn't have the same confounds or different confounds, or a way to poke at the system. I think is super valuable. This is exactly where we should be, whatever that intervention should be.
I've got
Well,
a wonderful colleague called Ekaterina, who was I was co-supervising her PhD, and she's now a postdoc, and she's done this great work where she's taken a load of different MRI measures and a load of different PET data, and is doing quite conceptually simple things, basically just doing kind of spatial correlations between these two different sets of data, but finding really interesting results, and correlating, doing things that people don't necessarily do, like correlating kind of cognitive behavioral measures with PET data and
yeah,
things like that. She's done some really great stuff there.
that's brilliant. So maybe I regret suggesting this, but I wonder well, first of all, where do you see these neuroplastic changes in the FA roughly? Are these are fairly localized, or are they very
Um,
it's a visual task, a visual motor task, right?
no. So the visual task is what we're looking at with EEG, right?
Oh, okay. Okay, yeah.
Sorry. With the FA stuff, it's just like standard
And
diffusion.
okay. And are they local changes? Just are they in certain regions that make sense, certain hubs, or brain wide?
Honestly, I struggle to remember. Sorry.
It's all right, no worries.
And
Because
I don't want to talk too much about this because it's
yeah,
not really my
yeah,
paper, and
sure.
it's not out yet. I hope
So
it will be soon.
so although this is a very tricky thing to do, I do wonder, and I wouldn't do the current standard approaches, but I do wonder if you were to do volume localized spectroscopy, if you have a good couple of candidate areas, especially if you have some control areas where you really are not measuring changes in the ligand uptake, then you could start to ask about cycling, GABA cycling, glutamate cycling. Even NAA level may change because, as I understand the location of NAA, it's mostly in the mitochondria. To me, it looks like an index of synaptic density anyway, right? The more mitochondrial chains you have, the more NAA you have.
Right.
Because it's in oligodendrocytes. Yeah, because it's in neurons and oligodendrocytes.
Oh.
So you would need a very good spectroscopist to figure this out. And I work with
Yeah.
one, and she would tell me, don't even think about suggesting GABA edited anything because she thinks it's a nightmare. But you could certainly just, if you get the right kind of pulse sequence, with the right RF pulses, what she does is she uses a lot of prior information. So she has 50 or 60 compounds for which she in test tubes for which she knows what happens to the resonances at different pHs and temperatures, and like a 3 by 3 matrix.
Right.
And then she does a forward model using some very crazy, complicated quantum mechanics, which came from solid state NMR.
Brilliant.
And so she can extract maximum information from a spectrum where you don't
Right.
need to worry about editing or anything like that. You just simply brute force the solution out. She can pull all kinds of things out. I'll gladly put you in touch with her if you're interested. You'd want to get a couple of decent spectra, not hard stuff. A couple of 5-minute spectra from a couple of locations, nice parts of the brain, ideally that you can shim well,
Yeah.
and have a look at it. See if
Yeah.
it makes a difference.
I've done a little bit of spectroscopy in the past, but it's always seemed like a bit of a dark art. And
You want to work with somebody
really,
who's a stormtrooper in it, right? You don't want to touch
yeah,
it yourself. I used to do this stuff. I don't touch it anymore. I talk to
yeah,
Lana. I let her do it because you've really got to be just off the charts good to make this work properly.
yeah,
But it's certainly something that as a next level, if you start to get a robust effect
mm,
in your synapse density, I'd ask the next question. Like, all right, let me just speculatively pick a few people, drop a couple of 5-minute spectra in here
yeah.
to see what we see.
And that's another nice way of trying to tie the levels together as well, if you like.
Exactly. Yeah.
The molecular kind of level with
Yeah.
the other stuff. Yeah.
You have a hypothesis, so that'd be super, super cool. Oh man,
Mm.
that's interesting. Yeah, good stuff. All right. Well, let me see what else I had on my list. Don't we blitz through everything?
How are you feeling about multiband these days?
Um, I think it's pretty robust. There's most people I think are following the rules, like
Yeah.
with respect to the multiband factor, total acceleration,
Mm.
given the coils that they're using. There are
Yeah.
some hangovers. We've still got some banding effects that are now, sort of at the
Yeah.
Prisma, in the XA version, XA30, there's definitely
Mhm.
visual banding. And Eddie Arbach knows about this, and he says that it's a nonlinearity in the transmitter. So this is a Siemens level problem, not something he can fix.
All right.
So you'll see it if you just throw a phantom in, or even a human brain, you'll see it in just the raw signal to noise. But when you run the stats on it, it doesn't seem to
Mm.
make a difference.
Mm.
So, it's a static artifact essentially.
Right. So we've just got a new Siemens Vida, which is quite exciting, and it comes with XA60, and it's
Is the
got
Vita the wide body one?
yeah,
Yeah. Okay.
it's got we couldn't afford the fancy the SEMA, unfortunately.
Yeah.
um, but it's got the Siemens multiband
Yeah.
stuff baked in now, and I've been testing it out a bit. It seems pretty good.
Yeah, I
Uh,
agree. Yeah.
I haven't. Have you? Is that
Yeah, I've tried both, and I
XA30 as
think
well?
so
Okay.
the way I think about this is, if you're doing sort of bog standard multiband, let's say you've got your 32 channel, your 64 channel coil, and you do multiband 3 or 4, and you're not doing anything particularly aggressive, like multi-echo or anything like that. Just
Mm,
stay with the product because it's
yeah.
pretty robust. I think the only time you should really be on the CMR side is either because for compatibility with multi-center studies, because your friends are doing it,
Yeah.
or you're doing something that's advanced. You want control of the RF duration, or you want to do multi-echo,
Yeah.
etc. Right? So I think you should ask that question first. But yeah, if you don't need any of the features in the in the CMRR, I would stick
I
with that.
haven't actually installed the CMRR ones on our new system yet, but I'm really keen to get them on there and do take the Pepsi challenge
Have you
between
done
the
any custom
team.
sequences yet on XA?
No, I mean we've just been testing. We only got this literally end of February, beginning of
Okay.
March, so we've been doing quite a lot of just figuring stuff out and
From my personal
testing and
experience, the
stuff.
biggest problem is just Windows 10, which apparently is also compatible. It's just this, like the security certificates, actually getting things on. It used to be quite
Right.
easy to do it yourself, but now that you have
Yeah.
to have so many security certificates, those are the things that get you. It's
Really?
real. It's a computing task more than a physicist
Right.
task now. But it's doable. I mean, and the
Yeah.
CMR help pages are pretty good about what's going
Mm. Yeah, modern versions of
on.
Windows, just a pain in the ass from around
Yeah.
them.
Yeah, the biggest thing for me still these days, my biggest reluctance for things like multi-echo is not multi-echo or
Mm.
per se, it's that we unfortunately still do need in-plane acceleration. to make it work. And there you do have that problem of mismatch between your reference data and your current volume by doing things like grappa, and nobody's come up with a good solution for that. You can approximate it using Fleet or GRE to get your auto calibrations, but I don't know that there's a perfect solution yet, a really good solution.
Okay, I wasn't really aware of that issue. So can you unpack that a bit?
Yeah, so if you're doing a acceleration factor 2 for grappa in plane, you first of all got to work out what you would use for the weights for the missing case baselines. So
Okay.
Siemens nomenclature, they call that the auto calibration scan. And in prehistory, it was always a single shot method. So they would use the delta K was 1 unit, right? So they would use a full K space essentially in a single shot. But that is intrinsically mismatched from a 2-shot skipped K space, because now the speed through K space is twice as fast as you need, right? So now you're going to get, in the case where you have no motion and everything is perfect, you'll end up with artifacts, ironically. So then people was like, all right, well, we'll do 2-shot. If we're doing 2-shot grappa, we'll do 2-shot ACS. Okay. But there, if somebody coughs or sneezes or swallows between the 2 shots, you corrupt the ACS and that corrupts the whole run.
Right.
Remember, like in the limit back in the day, I had a guy doing grappa factor 4. He was doing, this was for diffusion scan. So the TR was 10, 12 seconds. So he had a minute's worth of ACS. And what we figured out was the only way you could really do it was to get people to scratch their nose, swallow, do everything, and then start the scan. They actually basically asked them to do that, and then you collect the data, and then you just immediately look at the first images that come up in the inline display, make a go, no-go decision. Does this look, if it looks reasonable, you carry on. If it's not, you stop, you do it again. So it's kind of ridiculous. It's not very principled, right? But I think most people these days, if you have a good rapport with your subject and that they are not, I was going to scratch my nose. The scanner just started. I'll quickly do it now before, it's like, no, worst possible time. But I think if you're paying attention to the online display and you can see the data, you can make a pretty good, based on the ghost level. I always go straight to the ghost level. You can make a pretty good judgment on, is this really going to be stable data or not, good data or not. But it's just a shame that we don't have a good way to do multi-echo without requiring in-plane acceleration. Maybe someone will come up with one day. Or we'll just end up with faster gradients. Everyone's going to move to faster gradients.
Yeah. I mean, do you have to have grappa on to do multi-echo?
You don't, in principle, no. But
otherwise, it's just too slow.
Yeah, exactly. Yeah. Your TEs are too long. So most people grappa factor 2.
Right.
And now you get multi-band factor 4. Oh, okay. Now you're starting to really push things. My biggest concern is not the overall acceleration, but the motion sensitivity, or time since basis set collection. And we proposed this about, 10, 12 years ago. It's just to do multiple redundant basis sets and just make the decision afterwards. You can imagine collecting auto calibration scans, collect a bunch at the beginning because they're for free. Then your first TTL comes out and you start your script or whatever's happening and you run your experiment. At the end of the experiment, why not collect a few more and lock them away? And then you can ask the question, which was the best one for the total run post hoc, right? But nobody does that. There's no feature to do that at the moment.
I've never tried any multi-echo stuff, but the people who use it seem to be really keen on it.
I wonder if they'll stay keen if they start doing things like rapid tide correction. I wonder if they're actually just essentially using it as another way to get around global signal regression. The problem for me is the edge cases. There are, like, for example, the rapid tide, the systemic low frequency oscillations, they are a bold signal. So there's no good saying, oh, it's bold, non-bold. This is a legit bold signal. It's just not the neurovascular coupling that you're interested in. So you're already behind the 8 ball. So I think a lot of this is the way that you use the data. You can't just simply say bold good, non-bold bad. That doesn't work. But
one of my colleagues has just got a little grant to look at some obesity related stuff. So done a little bit of work with weight loss and obesity before, looking at mostly kind of, changes in reward circuits and that kind of thing when you show people pictures of food and stuff. But this group, they've just got a little grant to do some kind of methods development stuff to try and get reliable signals from the hypothalamus, which is a small area, really close to loads of airspace. It's like literally the worst, worst area to try and image. And another area called the area postrema, which is back of the brainstem somewhere, which again is really small. So I was thinking about trying out multi-echo stuff for that.
Yeah,
just to get a little bit more signal with the shorter TE down in those kind of areas. I don't know.
Yeah, worth a look. I would also suggest obviously cranking the resolution up as much as you possibly can to reduce the through plane dropout, or maybe even just doing thinner slices than you would otherwise, even if you don't get full brain coverage. And the definitely the look at the phase encode direction. PA may help you a lot there.
Oh yeah.
P, yeah, you might find that you retain a lot more signal. PA phase encoding, but yeah, I'd look at the multi-echo too. All worth a shot.
Yeah.
You can just look at just temporal signal to noise for these areas first, right? Just to look and see how as a proxy. That'd be good.
Yeah, it's not going to be easy to get reliable stuff there, I think, but well.
Yeah, tough. So what else, man? What else? So you're doing mostly chronic studies. No plans to go back. The acute stuff just too hard, or just not interesting scientifically.
Well, no, I mean it's just tended to be what we've done recently. So actually, it's not quite true. We have done in the last few years. We have been doing acute stuff with cannabis. We've done a lot of work. So there was a big project that I did with people from UCL where we were giving people fairly high doses of cannabis and putting them straight in the scanner. And hardly any of them freaked out, which was great.
Were they supposed to freak out?
No, of course not. But you expect a couple of at least, but no, they were excellent. They were very good.
And what are you trying to see? This sort of neural correlates of being high, just
yeah, bunch of different tasks. We got some spectroscopy data from that study actually.
Oh, okay.
And
is this like pure THC, or are you actually using marijuana strains that have CBD as well?
Well, so it's actually both. So it's two different strains of cannabis. One that is very pure THC with hardly any CBD at all, and another one that's a bit more balanced, and a placebo strain where all the cannabinoids have been removed as well.
Okay.
Yeah.
Interesting.
Yeah.
The paper might have been one of your papers about the CBD was important. You couldn't just assume it's all THC.
Well, there's been this interesting. There's been this narrative developing in that area that CBD is kind of protective against the effects of THC.
Right. Yeah.
THC is addictive and causes psychosis and so on, and CBD is kind of anti-addictive, anti-anxiety, anti-psychotic, if anything. So the idea is that we're getting increasing rates of people suffering problems with cannabis, like addiction and psychosis and so on, over the last 20 years. And because THC content has gone through the roof over the last 20, 30 years, you get some strains now that are like 45% THC, which is bonkers. Yeah. 20 years ago, it used to be 4 or 5%. Yeah. Now, commonly, it's kind of 10 or 12%. And there are some strains that will just, blow your head off. So yeah, there's this narrative that it's these pure THC strains that are really causing people problems, and that if people actually had a more kind of balanced strain with CBD and maybe some other cannabinoids in there as well, that would be better. And there's some data to back that up. I mean, I've got a couple of previous papers showing that you get strong disruption with the THC the pure THC strains, and a bit less disruption with the more balanced strains when you're talking about like resting state networks and things like that. What we actually found in the most recent study, which is I think this was actually a pretty big study. It was about 48 people. We had two groups, adults and teenagers as well, 16, 17-year-olds and kind of people in their mid to late 20s. But what we found really is that the CBD doesn't make a lot of difference. And if anything, it actually might make the effects of THC worse in this sample anyway.
So where did that narrative come from about CBD? Is this brought to you by the growers of your local cannabis?
Well, no, I mean there I
Smoke
think
natural.
I mean, partly it's just about that they seem to have kind of subjectively, they seem to have opposite effects in terms of, you know, THC gives you anxiety and CBD is anti-anxiety and things like that.
So self-reported, or is this in a sort of a population? This is in sort of a
well, no, I mean,
epidemiology.
kind of clinical studies where they've just given people and these things and got them to report stuff. And people have tried, you know, clinical trials with CBD for psychosis and things like that with, yeah, you know, some results. But I think it depends. I think it depends quite crucially on the relative dose of the two. So what we found, weirdly, this is really weird. We're giving people the same amount of THC each time, but if you look at the blood plasma levels, they were about 14, 15 milligrams per milliliter. No, that's not the right measure. It's about 14 or 15 of whatever units it was. I can't remember, nanograms per milliliter or something like that. In the pure THC condition, and it was about double the level of THC when you co-administer the same amount of THC with CBD.
Interesting.
And we don't really know why. We think it's because, well, this is our idea anyway. THC and CBD are both metabolized by the same enzyme, cytochrome P450.
This is oral, presumably.
It's inhaled. It's
Inhaled. Okay.
right.
Right.
Yeah.
So it's blood at this point.
Yeah. So we think if you get the right or the wrong relative doses, if you like, these two things are basically competing for metabolism at this enzyme. And the CBD is blocking the metabolism of the THC to an extent.
The area under the response is possibly the same. It's just that the time constant is now slow.
Maybe. Yeah, exactly.
Yeah, okay, that's interesting.
Yeah,
which still makes a difference, right? It's like eating before you go drink beer. Right?
Yeah,
slows down absorption in duodenum, so it makes sense, right? You have kinetics.
So CBD may have some protective effects. We don't really know.
Interesting.
But it may also, you know, increase at least the subjective effects of THC by that metabolic type method.
Yeah. It'd be interesting to see if the total effects end up lasting longer too, which would be interesting. I don't know. Probably, you know, if for people who are using this stuff regularly, probably they were probably never fully clear of the stuff anyway, right? Because they're doing 2-3
No, exactly. Yeah.
Yeah,
you don't know. Well, particularly, I mean, I guess it's probably better where you are because, you know, you can go to a shop and they have a menu and they tell you exactly what's in this stuff, right? I mean, whether that's reliable or not, who knows? But
I still think there's a lot of people still growing their own, and there's still a whole
oh, sure. Yeah.
But yeah, I don't know. I'm not in the space. Space adjacent. Yeah, it's fascinating. Yeah, well, that's good. So there's some good science questions to ask about that, and it's just certainly going to be important for a societal thing. We want to understand it, right? People need guidelines, right?
I mean, the whole point of this study was really to look at the teenagers aspect as well, because teenagers smoke a lot of weed. It tends to be the first thing.
Yeah, and the brains are still plastic.
Yeah, exactly. You know, and there's lots of rat work where you give, young rats big doses of THC, and then you find 6 months later that they have trouble running mazes and that kind of stuff.
I just want to sit and eat crisps on the couch.
Yeah. So yeah, I mean, there is genuine concerns
Yeah.
about teenagers, particularly teenagers smoking the really high strength skunk stuff that's around these days. In all the data that we've looked at from that study, we really haven't found much difference between the adults and the teenagers in terms of brain effects at all.
Interesting. And then in terms of like the enzymes,
Kind of weird.
are there genetic, are there phenotypes that would potentially mess up your data? You might need to do some genetics to see who has what levels of enzymes.
Yeah, possibly. So there's a gene called COMT.
Familiar
And
with it?
there's a really nice paper actually, which was just out about 6 months ago by some guys in the Netherlands, where they actually took a bunch of cannabis challenge data from a bunch of different labs. So we contributed some of it, and did a kind of mega analysis type thing and split everybody by genotype on this COMT gene, and found some quite strikingly different effects actually. Yeah.
Interesting. All right. So okay. So going back to the big picture for just drug studies overall, it seems to me like some sort of genetic profile of your subjects would make a lot of sense.
It'd be lovely. Yeah. I mean, there are, particularly when you're thinking about psychedelics, there are definitely polymorphisms of the 5-HT2A receptor that we think might be important.
Yeah.
Nobody's done a big enough.
Could be doubly important for us because there's cardiac, HT2B cardiac receptors. So we would want to know what parts, activating the heart versus the brain could be a big.
Yeah. That's a big concern actually in the people who are doing the kind of microdosing work. No one
might just be stimulating the heart. Is there a preferential uptake of one versus the other, or does it neatly divide 50-50? How does it go?
So I mean, they definitely, I mean, the psychedelics like psilocybin and LSD, they definitely have effects at 2B as well.
Yeah.
We don't really know what effect, that doesn't seem to do anything bad to the heart with, a single big dose, even semi-regularly, like, people do recreationally, We don't really know what a kind of a regular small dose will do. Probably it's fine.
You could measure heart rate and heart rate variability as that'd be
I mean, there's a possibility it could even be, cardiotoxic in some way and build up time. Yeah, we don't really know.
Well, yeah, I mean, I would think that like caffeine, I mean, we know there's receptors, we know that there are effects. Yeah, at some level it's toxic. Doesn't stop me from doing it a few times a day. But you would I definitely feel, you can certainly get into the point where your heart rate variability is, almost fibrillating, right? It's easy to get to that point. So presumably my heart rate variability is changing when I have my first cup of coffee. It wouldn't be particularly difficult to measure this effect if it's a robust effect in an individual with a microdose. So maybe you'd want an independent measure of heart rate and heart rate variability for each session. Just a couple of minutes of data for sure, if that can be collected. Yeah, are you familiar with? So I'm going to try and get Emily Jacobs on. She's running this new thing based here called the Women's Brain Health Initiative. So there are lots of genetics there too. So the idea is to sort of lots of imaging data and then lots of. Eventually, it's all bottom up right now, so everyone's just trying to get going. But I'm curious because I think there's a huge amount of overlap between things like big large N studies like UK Biobank, women's health studies, drug studies. A lot of this stuff is not just you can very easily get 5 years down the line and think, like, if only we'd collected blood data, if only we and it's too late. You've got to start thinking about this up front. And I think there's one thing that we've come up with is we don't have a registered nurse or someone who can do phlebotomy, someone who could do blood draws, but there's apparently these little patch things that are like micro they're like leeches. You pop it on, you leave it for a day or so, and it just sort of through
Really?
osmosis, where it collects enough, yeah, and then you take it off.
Oh, that's
And
wild.
right, and then you don't need any biohazard or anything. You literally
Right.
just like you do a stool sample for your doctor. You just it comes in a little thing. You drop it in the
In the
envelope, you seal it up, and you send it in the mail.
Wow.
Yeah, yeah, genius. So
Yeah,
I'll
yeah.
information on it. Yeah, yeah. We haven't got any yet, but we talked about getting those because that would
Mm.
the logistics is super easy, right? Then we can
Mm.
give somebody if they're interested in volunteering for a study, here, slap this on for a day, and then follow the
Mm.
instructions. And then
Mm.
we don't need any other special permissions or any special.
Doesn't need to be frozen or
No,
no, that's
apparently not. It's all they have whatever the chemicals are in there. They're
right.
doing the preservation in real time. You know, whatever the hemocid, whatever the
Right.
heparin or whatever is in there, and whatever
Mm.
it's doing it real time. I don't know if
Right.
it's just plasma or it's collecting red blood cells
Mm.
or not. I don't know. I'm not sure. I haven't looked
Mm.
into it in detail, but I understand that they are now available. Um,
Wow.
yeah,
Cool.
pretty cool. Yeah, it makes things
Mm.
easier. So
There's
I was going to ask you about the neuroplasticity. So the stuff that
yeah.
other ways of getting at neuroplasticity. You say, yeah, you've got your attention task. Your
Yeah.
other ways do you think that there might be at getting plastic to
Well, I mean, people have used things like, cognitive flexibility tasks and things like that. Um,
in behavioral?
yeah, in behavioral and in fMRI, um, doing those tasks and looking at the brain as well. That's one option. I mean, people say that these persistent changes you see in the resting state networks after psychedelics, people say these are, reflecting in some way this opened window of neuroplasticity. But there's a conceptual leap there, I think, that needs to be made. I
Yeah, you
don't,
could think about that. So the mechanism, right? So you could say, right, sure, but it might be a vascular effect. It might be through BDNF or other things that are upregulated. Or exercise does the same thing. Go run on a treadmill for 20 minutes. You'd get the same
yeah,
upregulation of potential benefits for neuroplasticity, right? It doesn't have to be a drug. So the real question is, what's the mechanism?
yeah, yeah. I mean, neuroplasticity is such a broad term, right? You can have, people use it in all kinds of ways, talking about, individual synaptic changes in sensitivity or upregulation, downregulation of receptor sites on a synapse, up to systems level changes, and everything in between.
Yeah.
So and it's not really clear, most of the time when people say neuroplasticity, particularly with relation to psychedelics, exactly what they mean. There's some stuff where it has effects on BDNF and things like that. So it seems to have a low level effect.
So it's probably just reflecting the individual's bias. They mean it to mean like Humpty Dumpty language, right? So if I'm a psychologist, I mean it to mean improved cognitive flexibility, brain wide overall performance.
Yeah,
If I'm a
sure,
cell biologist, I'm talking about synapses.
sure.
So yeah, probably the same sorts of things as go on with brain training approaches, brain training video games, where
Yeah,
uh,
yeah, yeah.
does it transfer and all the rest of it. Interesting. All right, well, cool. I don't know that literature. I'll look at that. I always think with any of these things, if you're going to, especially when it's something like neuroplasticity, I always think, well, why not have exercise as a control group?
Yeah, yeah.
Certainly with brain training, if you're going to spend an hour sitting fucking around with video games, maybe try the same hour walking around your local
Yeah,
park.
treadmill. Yeah, yeah,
Yeah, maybe there's a better maybe there's a and even if the overall cognitive benefits not dissimilar, you might find
mm,
the chances of liver cancer go down. So what's the downside?
yeah,
Unless you
yeah,
end up in the pub
yeah,
every night, you walk around
yeah,
the oh, I end up in the
yeah.
pub.
There's a guy I know actually at UCL who's just got a big grant, um, looking at the effects of exercise as antidepressant, basically.
Sure. I mean, there's a lot of
Yeah,
anecdotal evidence that suggests
yeah,
that it could be an effect, right? If it
yeah.
was patentable as a little blue pill, we'd have been doing it a long time ago.
Oh, yeah, for sure. Yeah, yeah,
Interesting. All right, man.
yeah.
All right. Well, any other final thoughts or questions? I'll let you go
No,
now.
this has been great. I thought you were going to kick the crap out of me for my terrible approach to
No, there's no
modern
point.
physiological noise.
There's no
But
point. And I'm gonna, I'm an equal opportunity, mean, nasty person. I'm gonna put Dan Handwerker on here soon when he gets back from OHBM, because I know he trained here with my ex-boss.
yeah,
So, and I know he's in the church, and the papers that he's been on, it's not his papers, but he's on them, and he's told me offline. Yeah, I see the same caveats. So I think where we are in this right now, I see people setting up to do a lot of this stuff. Right, we have our own psychedelics group now at Berkeley. I
yeah.
don't want to see people run into the same damn lamppost on the same dark street.
Oh yeah, yeah.
Why would I do that? I think we need to have a conversation now, and maybe people should just slow down a little bit and ask themselves the questions: what must I have now? What would I like to have? And if it's hard, okay, but I'm spending thousands of dollars on these things. Maybe I just bite the bullet and I do a pupillometry for arousal or whatever it takes. Maybe make it a little bit harder, but the benefits
Yeah,
huge down the road.
yeah.
I hate to see people just collect a bunch of data and then regret what they did.
There's never a downside to collecting more and different types of data when you're doing this stuff.
Yeah,
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 absolutely
yeah,
right.
yeah. All right,
Yeah.
mate. Well, good. Well, thanks for your
Cool.
time.
Really nice to meet you face to face, kind
Yeah,
of.
indeed.
Anyway,